So, okay, so for eukaryotic cells, last class you talked about epigenetics. So this idea that we have chromatin. Chromatin is when you have DNA plus proteins. And we have to wrap DNA around those proteins so it will fit inside the nucleus. Well, to access genes to do gene expression, to use those genes to make proteins, we have to unwrap that DNA from those proteins that organize the DNA to use it. And how we, the different chemical groups we attach or don't attach to those histones will change whether we express the genes or don't express the genes. So we had heterochromatin. So heterochromatin, that's when it was condensed, and that was a decrease in gene expression. And what kind of chemical group caused the heterochromatin? Anybody remember? Luke, go for it. Methyl groups, yeah. So if you see a methyl group, remember, don't do meth. Otherwise, you'll get less gene expression, maybe. Lower gene expression for if you have methyl groups. And then there's euchromatin, which is not how you spell that. Chromatin spelled with a K. That's an idea. So that'd be the opposite. It's uncondensed, which is going to correlate to higher gene expression. Now, understand why. Because if it's unwound from that protein, you can access the DNA that you need for that information you need to make the protein. And what chemical group would cause the euchromatin? I gave you a poker analogy. What card are you looking for when you're playing poker? Aces, right? So acetylation, an acetyl group, that will open up the chromatin, allow you to access the genes. And appreciate when we say access to genes, increase gene expression, what's really happening is you're making it more likely that the RNA polymerase and all the transcription initiation complex proteins can bind to that promoter region, and RNA polymerase can start doing transcription. So here's just a picture of that acetylation process. You add the acetyl group to the nucleosome. That's a collection of 8 histones. That's what you can kind of see there. So nucleosome and histones, similar idea. Okay, so getting into the actual details of how we regulate genes in eukaryotic cells, we will have what they call control elements. And basically control elements, they are non-coding segments of DNA. So a coding segment of DNA, that would be the literal DNA that would literally code for mRNA codons, where you could use the codon chart and see what amino acid that goes with. The non-coding DNA, that's they originally thought it was called junk DNA, which was basically they thought it was just some remnant of evolution and stuff. It didn't really have much of a purpose. But we now know the non-coding DNA, it really serves a major role in regulation of a transcription, along with some other things. But anyways, it's the important thing to understand, kind of bigger picture is we want precise regulation of gene expression because we're made up of different cell types. So what makes a nerve cell different from a muscle cell, different from a liver cell, is not the literal genome, the A, T, C's, and G's that make up those cells. It's how exactly you use those genes, the timing of gene expression. Okay, all right. So, kind of a lot going on in this slide, but let's walk through it bit by bit. So, here is our DNA strand. Okay. This would be our promoter region, that transcription start site. So, you should be somewhat familiar with that. We talked about that before. This would be what we call proximal control elements and distal control elements. I'll show you a picture on the next slide, a picture where we kind of use this. Basically, proteins are going to bind to these regions and either increase or decrease transcription. Okay. Now, transcription, that's the process where we take the DNA and we're going to make our RNA. Now, looking at our, and we call this pre-mRNA. You know it's pre-mRNA because you see exons and introns. Remember, it's backwards. The introns get removed. The exons, standing for the expression, those things that get expressed, They then go into the fully made mRNA. Looking at some of the portions of the mRNA that we've talked about, the 5' cap, notice that it's made of the guanine nucleotide, and then you have the poly A tail. There's also something called a poly A signal. Not as important, but if you're just kind of curious what it's doing, poly A signal, it really is basically kind of a signal that helps us know when we're gonna, like, where exactly we cleave that 3' end, and then also kind of helps signal like where we attach the things for the poly A tail. But thinking about the cap and the tail, there were two like major things I want you to know with the cap and the tail for why, like, the purpose of them. Anybody remember at least one of them? What do we do with the cap and the tail and the mRNA? Why have them there? Luke, go ahead. Yeah, good. Degradation. You can call it degradation. And then, plus you knowing that's a good buzzword, hydrolytic enzymes. So degradation by those hydrolytic enzymes. Hydrolytic hydrolysis, where you break a polymer into monomers. And then what's the second thing? And think of just like what needs to happen once you have the mRNA. Like, how do we do translation? Where do we do translation? The steps required to get there. Thomas? To the cytoplasm, yeah, okay. So how might the cap and the tail relate to cytoplasm? Because remember, we do transcription in the nucleus. Right? We're in the nucleus when we're doing this. But to do translation of this mRNA, we got to be in the cytoplasm. So the ribosomes are at. Transportation. The cap and the tail, they will help in, basically they'll provide some binding sites for other proteins to help, like, transport it out of the nucleus to the cytoplasm. Okay. All right, so kind of a lot of review stuff. So the next slide, I want to get into these proximal and distal control elements. First, let me make sure I said everything correctly. Yeah, so talking about these control elements, basically you have these things called activators and repressors that bind to these control elements. And these activators or a repressor, it's a protein that will bind to an enhancer. So the enhancer is this distal control element. So distal is far away. I don't know if some of you have done anatomy, but there's like proximal, distal, like as far as like if you're distal, you're really far away from the center of the body. If you're proximal, you're close to the center of the body. So proximal, you're close to the promoter. Distal, you're far from it. So an enhancer is this protein that binds to that distal control element, and it helps us start the transcription of the gene. Okay? Now, in order for that to happen, these activators, they have to have two domains. Basically two regions of the protein. One that can bind the DNA, can bind that that distal control element. I'll say CE for control element. And then a second that can activate transcription. So a second that can bind protein. Because this is going to need to interact with the transcription factors. Transcription factors, those are the proteins that will basically help get RNA polymerase to stick on the promoter so RNA polymerase can do transcription and make the messenger RNA. And that's what they're trying to say with the facilitation of protein-to-protein interactions, this interaction of the transcription factors and that RNA polymerase. So let's kind of look into some of the, well, first let me show you the actual structure of an activator. So just trying to show you the two different domains. So you have a DNA binding domain. So this would be here. This would typically be the distal control element. So know that region, not the proximal. Typically it's the distal control element where the enhancer is binding. And then in the activation domain, this would be binding a protein. Okay? So in other words, the chemistry of like this part of the polypeptide and that part of the polypeptide are going to be a little bit different. The amino acids that we need to make up the DNA binding and the activation domain are going to be a little bit different. All right, so let's get into the details of this. And I think this is really cool, and it follows. It looks very overwhelming at first, but hopefully you'll see it actually follows some common sense for like how this. Once you I think kind of see this, actually it makes sense that it might work this way. So here is our promoter region. That's where the promoter again. That's where the RNA polymerase binds. RNA. That's where the RNA polymerase wants to get bound. Okay. Now, remember the ta-ta box? The RNA polymerase is looking for the ta-ta. Okay. Ta-ta box. Now, there's the distal control element. Here, what they're not showing is the proximal control element. Whenever we're like enhancing gene expression or repressing it, the distal control element plays a much bigger role. Proximal control element will bind some other transcription factors, but focusing on the distal control element, you have these things called activators that bind. And they bind to what we call an enhancer region. So what is it? What is the enhancer enhancing? It is enhancing gene expression. What are the activators activating? They're activating gene expression. So the name tells you what they do. Now, distal control element. It's called distal because it is far away from the promoter. So if you're thinking critically, you should have thought, how exactly does a protein binding there somehow turn on this gene? That just seems really goofy, right? It's like me pressing a button way over there and that's somehow turning on these lights, right? That should like kind of, like you shouldn't just like be like, yeah, okay, that makes sense. Like there should be some way that those relate. The kind of the connector between those is the DNA bending protein. So this is kind of wild. You will have a separate protein that binds to an area in between the distal control element and the promoter, and it will do exactly what it says. It bends the DNA, and it'll bend the activator proteins so that they can meet up with the mediator proteins. So in other words, you have an activator, and there's a middleman between the activator and the transcription factors. Transcription factors. You have an activator. Activator connects with the mediator. The mediator mediates the meeting of the activator and the transcription factors. The transcription factors, they are what directly allow RNA polymerase II to bind, and then boom, you start RNA synthesis. This whole complex here, this is called the transcription initiation complex. Because it is a big old complex, right? There's a lot going on there. But I think the names do actually make some sense, right? Activator is going to activate. It gets things going. The mediator mediates. You have a mediator in an argument. They're like the middleman. They're trying to promote civility. The transcription factors, their job is to give a spot for the RNA polymerase to start. Because RNA polymerase, this is important. I mentioned it when we were doing chapter 14. RNA polymerase can't initiate its own transcription here. It can't just do it on its own. It needs a bunch of helper proteins to make this happen. Okay? Any questions about this? Okay. I think it's kind of neat. Now, this slide is really more or less the same thing, just kind of another picture to sort of help it make sense to you. So, the right portion of the picture is a little bit different, but the right portion is just showing here's your enhancer region of DNA. So this enhancer, this is part of the distal control element. So in other words, if that sequence of DNA on the distal control element is going to end up turning on, it's going to end up raising gene expression. We call that region, that part of the DNA, the enhancer. This right here would be your activator protein. Okay, and then here's the DNA binding protein. These would be your other transcription factors. Now they're not showing the mediator proteins, and so be it, but kind of and here's a ta-ta box. So RNA polymerase, and they're showing a green check mark. That's showing that gene expression is happening. Now, there is then the opposite thing where we want to turn off gene expression. We want to lower gene expression. And it's a similar mechanism, but instead of it being the distal control element being an enhancer, now the distal control element is what's called a silencer, because it silences gene expression. It silences transcription. So this will bind then to what we would call a repressor protein. It's a repressor protein because it is repressing gene expression. Specifically, it's repressing transcription. Okay, so now RNA polymerase can't bind there. So notice how that's the dashed line is saying that RNA polymerase is not there anymore. Okay, questions about this? And don't forget the DNA bending protein. So there you go. So let me make sure I said everything here. Yeah, and what can change from gene to gene is like where exactly the transcription factors need to bind. As you can imagine, like this wouldn't be like that process there can vary from like gene to gene, from species to species. But the basic idea would be it would be about the same idea, having this kind of bending of the DNA and all that. Oh, okay. This is something interesting that I haven't mentioned. So let's say you have there's no enhancer. Okay, so no enhancer, meaning no activator protein. The question is, well, is there no transcription of that gene? And the answer is no. There is still low rates of initiation, meaning low rates of transcription. So low gene expression. And that's because you still can have those general transcription factors binding and helping the RNA polymerase get going. Kind of a subtler point, but I think it's important to make note of. It's not, in other words, it's not all or none. It's a little more of a dimmer switch. The enhancers are going to just, again, they're going to enhance. If I want to completely shut it off, that's when you have the silencer. Okay? Or you could then attach the methyl groups to the histones around that portion of the DNA and just wind that DNA back around those proteins, if that makes sense. Okay. So enhancers increase transcription, silencers decrease it. Another kind of lower key point is that the location of the distal control elements, it could actually be part of an intron of a different gene, which is kind of interesting. So sometimes introns of a separate gene could be an enhancer or silencer for another gene, complicating things even more. So the picture I showed you was showing direct control of the activators and repressors to like turn on or turn off gene expression. But these activators and repressors, they could also kind of do a similar thing here where they influence the heterochromatin or euchromatin, whether the DNA is bound or unbound to the histones. The details of that you don't necessarily need to worry about, but just kind of be aware that, again, this can be a little bit more complicated where some of these activators or repressors, maybe they then recruit other enzymes that maybe attach methyl groups or acetyl groups depending on whether you want to turn on. So in other words, maybe an activator could recruit other enzymes that attach acetyl groups, and the repressor could recruit enzymes that are attaching methyl groups to the histones, so winding up the DNA or unwinding the DNA. All right, now this slide I think is actually really kind of interesting. So we talked about different cell types, right? So you have here they're looking at, hey, let's say you have a liver cell versus a lens cell. They all have identical genes. How exactly would the liver cell specifically activate one gene and the lens cell another? Because the question could be like, does that mean like we have like a gajillion different like activator and repressor proteins, and that ends up getting, and they have their genes that like code for them, And you can see how it can get very complicated very quickly. But what we actually have is, bless you, we have very like common, think of like keys what the slide is trying to get at. Like the key for my door and the key for like the math hallway, they look very similar. It's a similar concept, but they will vary slightly in like the positioning of like in the depth of the grooves on the key. So what's going to turn on, say, the genes that we need in the liver cell? So in this case, we want albumin. Albumin's a really important protein in liver cells. It'll help keep blood volume up and bind certain hormones in the bloodstream. Don't even know the role of it. But crystalline, we want crystalline to be expressed if we are a lens cell. Crystalline will help form the lens of your eye. So if you're a liver cell, you don't need to make the crystalline protein because you're not trying to make a lens. But you do want to turn on the genes again for albumin. So here's how it would work. The liver cell and the lens cell, they will have different combinations of activators. Different combinations of activators. So they call this concept combinatorial activation. So activation, turning on a gene, how we turn on that gene is based on the different combinations of these control elements. Okay? So in other words, if I want to turn on the gene for albumin, I need this funny looking, and that funny looking activator. Okay? So different combinations of activators. I need these three activators present in a liver cell to turn on the albumin gene. Now, in the lens cell, the lens cell will have this silver looking one, right? But because it doesn't have the this orangey looking one and the yellow one, it can't turn on the albumin gene. It can't activate that albumin gene. And then vice versa with the crystalline gene. To turn on the crystalline gene, you need the pink one and the gold one here. So yeah, liver cell, it's got the silver one, but it doesn't have the pink and that goldish brown one. Okay, so I think the idea, again, is actually fairly simple. It's not much more complicated than just different keys. Okay. I got a question for you. What did this symbol mean? Not as important, but just something I haven't mentioned today. What are they trying to say with that symbol? Let me give you a hint. The enhancer is at the distal control element. Distal means far away from the promoter. Thomas? Are you asking, does this represent the proximal control element?
Is that just saying, like, there's more space?
Yes, it's saying this is far away. Like, for space, like logistical purposes, they're showing it to you close together, but they're trying to say this space right here, this could represent like 20, 30,000 base pairs of DNA away. Right? So then that's what you're seeing here, right? The bending here. Now, vocab, what would this be? What would you call that? Its name is literally what it's doing. What is it quite literally doing? Luke? It is bending DNA, so we call it the DNA bending protein. Thanks. Okay? What are these? Their name is literally what they're doing. What are they literally doing right now? They are mediating. They are the mediators. They are mediating the activators so that they can bind to, what are these called? The general transcription factors. The transcription factors. So they connect the activators to the transcription factors, and then the transcription factors, they will turn on the RNA polymerase II to do transcription, to turn on that gene. Okay. Questions here? I know it's kind of a lot, especially Friday before spring break, but make sure I said everything here. Yeah, so again, you can maybe only have a dozen control elements, a dozen different shapes of those activator molecules, and that 12 different. Think of like, Sonic. We don't even really know Sonic. Like the different combinations of drinks at like a smoothie place, maybe. Like you have just 12 different ingredients that actually can make a whole bunch of different combinations that we can use to turn on and turn off genes. Is what they're trying to say. This bullet point is what I was saying it even before I started recording, that the operon concept, where the genes are organized, that only applies to bacteria. We don't have operons. We don't organize our genes. Instead, we use those distal control elements to then bend and turn on certain genes. Okay? And this is important. Each genes that work together, they have the same combination of control elements. It would be like all of the rooms in the science hallway, they all have the same locks. They all fit the same key. They can be located in different portions of the school, but they all have the same lock. Okay? These genes that work together, they have the same control, the same lock. And that's beneficial because you want them to respond to the same environmental signal. Okay? You want them all to work together, kind of like what we saw last class with the operons and like lactose being present or not present, or you needing tryptophan or not needing tryptophan. Okay. Almost done with this part. Yeah, and the purpose of all this is, again, to fine-tune gene expression to changes in the environment. If a eukaryotic cell, you're getting a lot of different signals coming to you each day, or every second of each day, and so your cells may be able to rapidly respond, turn on and turn off genes depending on what's happening in your environment. So another way we can control gene expression is by alternative RNA splicing. And this is something we've actually talked about a few different times, so I'll go through it quickly again. That's just where we change what is going to be an exon versus what is going to be an intron. Okay, that will then change. That then changes what you then get different polypeptides depending on what you call an exon versus what you call an intron. Okay, meaning it gives you one gene can code for many different proteins, so it makes it more efficient. You don't have to have it decreases the number of genes you have to have in the DNA. And that's what you can see here. So here's a gene for troponin. Troponin is it's used in muscle cells, so it's really important for muscle cell contraction. But the specific form of troponin that you have in skeletal muscle, that's in the muscles I'm using right now, versus heart muscle or smooth muscle, it's in your digestive tract, or that's muscle you don't have conscious control over. Each of those cells will make different forms of troponin. But it's the same, they come from the same gene. Okay, they all are using the same gene. The difference in that the actual final form of the troponin is how you do the splicing of the RNA. Which, so maybe if you keep 1, 2, 3, 5 as introns versus 1, 2, 4, 5, that changes subtly the structure of that troponin. So it's really kind of clever how eukaryotic cells have that set up. Okay. All right. So what I've been talking about so far is just is regulating transcription. We talked about regulating the chromatin. We can regulate gene expression by regulating transcription, but we could also change gene expression by regulating translation. Specifically, we can make mRNA, but let's say all of a sudden the conditions in the environment where we needed that protein, let's say all of a sudden we don't want that protein present, but the issue is we already did transcription and made that mRNA. Well then, what we would want to do is we want to block translation. We want to shut that down. You don't want to make that protein. Maybe that could then be toxic to the organism. So we can block, using certain regulatory proteins, we could block, bless you, where exactly on the RNA that ribosome would bind to start translation. We could also turn on translation of all of the mRNAs all at once. And this is actually what you see in fertilization. So when the female makes her eggs, she'll have a bunch of mRNAs primed and ready to go. They're just waiting for the sperm to come, and as soon as that sperm fertilizes the egg, boom, all of these translation initiation factors, all these proteins, will then turn on translation of all those mRNAs that are waiting in that egg so that that embryo can start developing. Okay. Another way we regulate translation, or I guess translation, but just like gene expression as a whole, is the lifespan of mRNA molecules. So think about it. mRNA, it's not going to last forever. If let's say if eukaryotic cells, they typically have mRNA that lasts pretty long, and that's beneficial. Think of like the egg example. It's more efficient to kind of keep like a storehouse of potential mRNA in case all of a sudden you really, really need a bunch of protein. You could then start doing a translation. And kind of a side note, in that untranslated region, part of that region of the DNA that's non-coding, doesn't code for a protein, that's part of what the UTR can do. It can have, that region can kind of help promote the lifespan of that mRNA. Okay? But again, just understand this idea that the mRNA molecules have different lifespans, depending on like how quickly or not quickly the cell needs to respond to making protein. Okay, we can then regulate gene expression after we've already made the protein. So again, imagine, all right, the cell thought it needed a certain protein, needed a certain enzyme to do a chemical reaction, but all of a sudden, life changes, the environment changed. Well, maybe we then need to mark that protein for destruction. So we could tag that protein with something called ubiquitin. So I want you to know that name, ubiquitin. It's a little marker you put on proteins that marks them for destruction. Who do you think destroys them? What part of the cell? Think back to your basic cell parts. Get to you got it?
I was going to say phagocyte. That's the whole cell. Let's see. Yeah, it's the lysosome, right?
Actually, as I was saying, it's actually something called a proteasome. It's actually a completely different part of the cell. I was going to say lysosome was what I was thinking of, but then I was like, oh, it's actually not completely. Proteasome is kind of like a lysosome. It's like a similar idea. Anyways, the point is, I want you to know ubiquitin. That's what you attach the proteins to mark them for destruction. We don't want to do this, to be clear. This is not efficient. This is like hey, oh my gosh. This is again, something changed in the environment that the organism's in. We need to change how we need to rapidly change. We can't wait to just turn off genes. We need to literally destroy the proteins we already made. Okay? Or maybe we regulate just the length of time of it. Maybe you, anyways. We also, and I've mentioned this before, we will do certain cleavage and chemical modifications of protein that will control where they go. Not quite as important in this conversation, but maybe a certain part of the cell really needs the protein more than another part of the cell. Another way we regulate gene expression is by targeting that protein to maybe a specific part of the cell, or maybe specifically to export it to another cell type in the body or something. Maybe a certain organ in the body is really, really under duress, so we want to target that protein to that organ. Okay, let's take a little break. I know that was a lot.